Hello, and a very warm welcome to everyone joining us for today's Select Science webinar, How to Avoid the Next Nitrosamine Crisis, 5 Lessons Learned about Impurity Detection. My name is Sarah Thomas, and I will be moderating today's presentation. I'm delighted to be joined today by Heather Longden, who works in Regulatory Intelligence in the Pharmaceutical Marketing Department at Waters Corporation. Heather is a specialist in compliance to e-record regulations, acting as a resource to the global informatics community at Waters and to the users of all Waters informatics products. Today, Heather will discuss how analytical data can help to support confidence in the quality of manufacturing and how this can help to avoid contamination issues. The webinar will cover key analytical tests, how to improve methods and gain more information about your samples, and how Waters' analytical expertise can help your scientists and chemists support the quality of your food, cosmetic, or drug products. After the presentation, we'll move on to the question and answer session, so please feel free to ask any questions at any time during the webinar, and you can submit these to the left of your screen. Just click on the purple tab. So without further delay, I'd like to hand over to Heather for today's presentation, and I'd like to thank her for presenting to us today. Please go ahead, Heather.
Thank you very much, Sarah, and thank you for having me as part of the webinar series at Select Science. As Sarah said, today I'm going to be talking about analytical methods for detecting impurities because we've had a lot of information in the news about nitrosamine impurities found in certain classes of sartans and also ranitidines, and I'm going to use that as a case study for what could we do to avoid whatever the next nitrosamine crisis is going to be. I'm asked that question a lot, so this is really looking at the lessons we can learn from that to avoid that kind of crisis happening again. I'm going to talk a little bit, just a background about what that particular impurity was and how it was found, and then look through 5 key lessons about how we can have our culture of excellence and data integrity, and when we're looking at the data that's being generated by our QC labs, how we can automate that as much as possible because that allows people time to really look at the data. I'm going to look through the analytical methods, what you'd expect to be in them, both from a technology point of view and a scientific point of view for looking at impurities, but also how to improve those analytical methods in order to avoid mistaking impurities for other kinds of peaks in chromatograms. And then also automation generally, how we can improve processes in the laboratory. And then going to talk about a few extra considerations we need to take about supplier lines and about incoming materials and why it's important to test in those areas. And then we'll look at the bigger picture of how we have to look at our processes, and then from those we can design appropriate and fit-for-purpose analytical tests. So what are nitrosamines? What are the issues? And this has really been going on for about 18 months, maybe a little longer, when we first heard reports about nitrosamine impurities, first of all NDME and then NDMA, being found in commercial medicines in the mid-2018s. Then as things went through, we can see many press releases, especially towards the area of September last year, where both the FDA, the EMA, and other regulators really started to say to all pharmaceutical manufacturers, you know, this is a problem. What really happened there? Well, when a change in the manufacturing process was used in the creation of the sartans, particularly initially valsartan, to using a new solvent in the manufacturing process, DMF, it was found that that new solvent could react with the product and produce nitrosamine carcinogens. They were detected by a company who'd purchased from another supplier an API, and that was traced to production of these impurities in the new manufacturing process. And normally, an incident like that could be isolated, and those materials that could be affected could easily be taken from the market. But unfortunately, this contamination spread because that same solvent that had actually caused the creation of those impurities was recycled and then sent out again to other companies. and other companies who maybe were using that same solvent, which was contaminated with the nitrosamines, were using that in their manufacturing processes. And so the presence spread through many different products who were involved in and touched those materials. So it's not specific to the sartan manufacturer. It actually could end up in many other manufacturing products through a contamination. Now, while we were looking at the NDMA and NDEA impurities in sartans, it was also found that there was a reasonably high level. In fact, in some cases, it was thought to be a very high level of NDMA in ranitidine. And the source of that seems to be very different. So while we started to look for these impurities, now we're finding them in other products, maybe not even related to this particular contamination issue. So anybody who's involved in the pharmaceutical manufacturing process really needs to be aware of what's happening. Any companies or contract manufacturing operations that are either making the API or buying API or even other raw materials from sites that might have been contaminated. This means really rigorously testing all incoming raw materials as well as any manufactured batches or products that you're creating yourself. The FDA expanded their own testing to see if they could find these impurities in other products as well, and are also expanding on test inspections of manufacturers. And anybody who's supplying product to the European organizations needs to do a risk assessment in just about a month's time of any manufacturing process where these could be at risk. So it's creating a real big problem. It's probably creating shortages of drugs. Companies are spending a lot of time remediating this, and really this is something that should have been avoided, which is what really I want to talk about today. And we're going to look at these 5 lessons about data integrity, methods, and two lessons to learn about methods are blind spots, and then critical thinking to avoid this in the first place. So let's start with that data integrity. We want to be sure that the processes that we have in place ensure that our peer review, our lab review, and our quality review would identify any potential contamination in incoming raw materials or anything that's coming as a product that we're making. And I'm going to talk a little bit about how we might help to address that area. We need to ensure that when people are reviewing data that they have the time to do a real critical review, and any automation you can do in a review process or data integration or processing means less time checking manual data entry. Quite often these things are really very subjective to how the data is processed and how the data is collected, and the analysts do have to intervene, and the more the analyst has to intervene, the more somebody needs to be checking that the analyst intervention was correct. And really in a review process, we're looking for trends or outliers, and we're trying to track any manual intervention in the data which might have been, specifically in the case of an unexpected impurity, which might have been obscuring something that really shouldn't have been in the product or in the incoming material. And we need to be able to identify what's a real exception, something that is a real product issue, versus what is something where an analyst is just trying their best to get the job done. And when we talk about chromatographic peak integration, that's kind of key. Additionally, those analysts and the reviewers need to have the courage that if something looks suspicious or you're not confident in what that data is highlighting, then it's important that you can raise a hand and say, I think there's a problem here. Let's investigate it before we ship any product or use that material in our own testing processes. Now, just last month, a warning letter came out which really highlighted all of these. So I was going to not include a warning letter in this presentation, but this was just very much in line with the topic of the conversation. Now, first of all, there was failure to properly investigate a foreign particle contamination. It looked like the product was simply filtered and then shipped out without looking at the root cause of that contamination. There were some issues about GMP lab data, which is really the topic I talk about most. And in this one, the QA reviewers had full admin rights, which meant they could potentially delete data. They probably didn't, but there was the potential. And there was a number of standalone systems in the laboratory which were missing Part 11 controls like audit trails. But what caught my eye was there was also some concerns about the chromatographic analytical method, and particularly about peak integration. And it was really calling out a failure to properly integrate co-eluting peaks during impurity testing. And the result was product which actually should have failed an OOS specification and be an OOS result was actually allowed to be passed and pushed into the next line of manufacturing or the shipping, I'm not sure which. It's very important when we're looking at these impurities that the test is there to actually look for something that shouldn't really be there. And the people in the lab are under pressure to get results out and get ship product. And whether their errors are through inadequate peak integration or just inadequate methods, it could result in products being shipped with higher levels of impurities than they should do. Equally, it could end up in being product which is not shipped and is stopped shipped and is held when the levels of impurity were actually not that high, so it could go both ways. So when we talk about data integrity, often people think I'm talking about just the computer software, and really what I want to get across today is that it is a lot about people as well as the methods and the back-end IT parts as well. And we talk about how people have governance and quality and data review, and that's one of the topics we're going to talk about today. But equally, the actual method needs to be a very high standard, including system stability tests and good method validation. And even the science behind the method needs to be good. When we look at the computerized lab systems, yes, we do have to have built-in data integrity controls. We do need to have it validated, but equally, we should be able to automate as much as possible to minimize the human intervention, which is where bias can come in when looking at analytical results. And then we need our IT people to be sure that they're capturing the data, because we may need to go back and look at that data at some point in order to see, well, if there is a contamination, how long has this contamination been happening? How long has this impurity been formed, maybe in stability studies? So what are the signs when people are looking for data integrity concerns in the lab, and what is normal expected behavior? Because lab processes are generally not fully automated. You will find errors and mistakes. That's part of the reason we have a review process. But we see a number of trends that are coming when QA people are looking at the processes in their lab. And one of the concerns is whether they are looking at all the data, or are they simply looking at the data that's been submitted to them as final? Was there information in failed results, for instance, which could have been very important? We're also going to look at suspect data. And again, when you look at the actual analytical data that's generated and the results, there could also be very many flags, red flags, that might say, I'm not sure if this is quite the right data. I'm not sure if this is hiding something really important that I need to know about. And that's when you see things about peak integration, processed too many times, manually integrated, or very complex. And then finally, we see a lot of talk about audit trails and about the information that an audit trail could tell you that might also suggest that some process in the lab wasn't being followed according to the SOP. And I'm going to talk first of all about the incomplete data. Quite often, the data is collated by a lab and is sent to QA, and how do we know that the results that they are reviewing are the only version of results that were created? And just as an example, I pulled up an example here from what could be happening in a chromatography lab. Ideally, somebody would just run the samples, they'd get a result, and they would report that result. But unfortunately, in reality, they may have to actually repeat the analysis if something goes wrong with system suitability. You can't accept data from a system that's failed system suitability. And then potentially, they might repeat it again. And this is a trend that we need to be looking for in the data. Traditionally, if we do process the data multiple times or acquire it multiple times, we only reported the final result. This was the one that was deemed to be correct and acceptable. But those other versions that exist may be hiding people tweaking with some values in order to make the peaks bigger or in order to make the spectra fit exactly what it's supposed to in the SOP. So having some process that is routinely looking at that data hopefully is saved, but having it reviewed from time to time to see does this superseded data show anything that we maybe should have been aware of before we approve those final results. And really that's what I mean by talking about orphan data. It's that data that didn't get reported. So now let's have a quick look at the suspect data. And I think this is where, in that warning letter we were looking at before, they were talking about how peaks had been integrated and how they had been reviewed and whether the review had seemed to be correct, whereas the auditor that went in thought that the integration wasn't correct. And you can see that there are tricks like looking for manually integrated peaks, looking for data that's processed multiple times, but also looking at very complex integration parameters. Additionally, one of the most suspect ones are the results which pass the specifications so they can be shipped, but they only just pass by a whisker. It could be that result was massaged into passing the specification. So looking through that orphan data is often very important. And really, I want to look at some of the causes of that. When we ask why people have to do manual integration or process multiple times, we have to actually look at the data that's being created. And so this chromatography data shows unresolved peaks. And you can see that a software such as Empower that we used here today may interpret those peaks and integrate those peaks differently, and that's under the control of the analyst. Now, which one is right is not something I can really say. It all depends which peak we're looking at. The one that is resolved at the end is integrated the same in each process, but the peaks in the group are integrated very differently. So analysts need to know which is the right way to integrate those peaks. And that will be the way that it was done during the method development and the method validation. And it doesn't really matter what parameters the method has, as long as the outcome of the integration looks like whichever choice has been deemed to be the right one. That's a fairly simple chromatogram, but you can get very complex chromatograms. This is a chromatogram of a blank injection, and you can see the blank injection has many peaks in it, and it has a very strange baseline. Trying to accurately integrate real component peaks on that particular method is going to be very difficult with chromatograms like that. And if I show you what the injection looked like, you can see here that it may be very difficult to work out where the baseline should be on those blue lines in that section there. However, you can see the peak that is halfway up the slope is very reproducible between the sample and the blank. That peak should not form part of the sample analysis. It should be excluded because it's obviously not something that's coming from the sample. It's something that's coming from the mobile phase or the system or the diluent. So knowing exactly which peaks are impurities, which peaks are blank peaks, which peaks are system peaks is part of the job of the analyst, and their skill is required in order to process data like this. So there are regulatory concerns about suppressing peak integration because maybe the peak you've decided not to integrate is actually the one that is the impurity that you're supposed to be monitoring. And knowing when you have non-impurity peaks like the one I just showed, that whether they come from the system, the excipient, or from the diluent, is really important. And traditionally what people would do is not integrate those peaks so they wouldn't show up in the calculation. However, if we know that that's a diluent peak or we know it's something that's not an impurity, labeling it and then excluding it from the calculations, as you might be able to see in the little screenshot, we can label peaks that should be excluded from our total impurity analysis. We can still integrate them, we'll record them, but they won't be included in final calculations, and that seems to be a much more transparent way of processing the data than simply inhibiting the integration or making those peaks not be integrated. Even if the chromatography system finds those peaks and integrates them, if you have a complex and manual way of collating those data and submitting it to your quality people for review, undesired peaks or unexpected impurities could easily be missed. And especially when those reports are being collated into common office tools or if they're in a LIMS system, it would be very easy to just skip a line and then that would not show up in final reports or certificates of analysis. Anywhere where you've got what my colleague calls stick-aware, which is when analysts are typing data from one system to another, errors can happen, but also there could be deliberate exclusions of data as well. And then I just want to have a quick look at audit trails. I mean, audit trails will often tell you many things about what was done in a software. whether it's data that's been moved or copied or deleted. Hopefully nothing is ever deleted, or changes to the metadata that we wanted to take account of and make sure were done appropriately. However, I want to just explain that when we're looking at audit trails, you're only seeing a part of the story. You're not going to really see keystroke action or everything that was recorded on the screen when the user was using the software. But anytime they change or modify or delete a record, that would get recorded. If we look at the audit trail review, these are the quotations from different regulations that talk about the readability and review of the audit trails, and that's from the European regulations. For any change or deletion of any GMP data, the reasons should be documented and should be converted to a generally intelligible form, and then those three little words at the end, which was "and regularly reviewed," which made everybody decide they needed to look at the audit trails. How often and who was part of the FDA guidance. It talks about whoever's recording the record should be recording the audit trails at different stages in the process, probably maybe after each significant step, but definitely before you release a batch. And understanding what data you're looking at, what the audit trail tells you, will say how important the audit trail review would be. In the MHA guidance, it does specifically call out that audit review is not expected to implement a forensic approach to data check-in. It's not necessary for audit trail review to include every system activity, but routine data review should include a documented audit trail review, and it needs to be somebody who understands what the audit trail would mean. And how do you document you've done that? Well, the World Health Organization guidance talks about that as being risk-based. The roles and responsibilities should be assigned, and documenting the review is typically done by electronically signing the electronic data to say that you've reviewed and approved all the data, including all the versions we talked about, but also the audit trails. So when we're looking at it, you can hear that theme in there that says looking at the audit trails while you're reviewing the data is really very important. And this example should explain why that would be. And this is just with a Word document with track changes. If I only look at the changes on a Word document, I can't really tell very much. I can see that mistakes were made, and I can see what was crossed out, and sometimes I'll be able to see what has been added into there, but I can't really understand what this document is talking about by just looking at the changes. If I look at the final result, the data here, now I can't see the changes. I can see that everything is good, and I could look at all those numbers and go, yes, that's great, but I don't know what previous values were. I don't know which parts of this have been changed, and it's only when I'm looking at the two things together that I can actually understand how this document has been changed, and I could approve whether the changes were okay. And that's really the same thing we're saying when we say when we want to look at the data, we should be looking at the audit trails at the same time, because that will give us the confidence that the data can be trusted. For instance, in our Empower CDS at Waters, we have a number of different kinds of audit trails, a system-level audit trail, project-level audit trail, methods, which have their own audit trails, and then we have the story of all the results. Every result is saved and linked to the other results from the same injection. And that story, together with the sign-off and the sign-off reports and exactly what data people looked at when they were doing the approval, gives us confidence through the process that everything was done according to the SOP. But trying to say to somebody, now just go and look at the audit trails and tell me if everything is good, is not very helpful without actually looking at the context of those audit trails and using it in relation to the data that's being reviewed and approved at the same time. So a quality review process should be documented, but it also needs to be performed by a subject matter expert, somebody who understands the SOPs, the systems in use, and what the data would do. You could have a very good quality person, but if they don't understand laboratory processes, they're not going to be very adept at spotting unusual things in laboratory data. And we should always be looking at the original electronic data, not the static paper data, because a printout is just a snapshot of the results in a moment of time and doesn't necessarily tell the whole story. And not just look at that one data set that somebody asked you to review, but see if there's other data related to those sequences. Where it's appropriate, or include the audit trails in the review, especially where manual intervention has been made. And any exception reporting that you can do to try to focus the reviewer on the specific data that really needs to be looked at in detail and making sure they have all of the data to look at. So that's what we see when we talk about data integrity. It's those kind of processes in relation to lab processes. But what's the reason? Why are analysts struggling to integrate peaks correctly? And a lot of that is because methods tend to be very static once they're implemented. When we look at analytical methods, specifically around impurity analysis, sensitivity is so key because we're looking at very low levels. And as technology improves and as methods improve, we can see lower and lower levels of these particular compounds. And this is one of those things with the nitrosamines. Typically, people use the GC to look at them. They may have used a single quad mass spec on that GC. But when we use a higher sensitivity equipment, we can see much lower levels. And I think that's part of what's happened with the nitrosamine story. Robustness is always really important, and especially when you have lab people who are having to adjust results to get them accurate. Any robustness that we can have in the method to make sure that the peaks are integrated in an automated way is really going to reduce the out-of-spec results or abnormal results that people have to spend more time looking at. And then we need to look at methods. One of the key things about the NDMA and the nitrosamines is that most of the time people were not really positively looking for those compounds. If they didn't expect to find them, the analytical methods were not designed to find them. As with anything, analytical methods are designed generally to look for what you tell them to look for. And if you're not looking for a particular component, it probably may not show up. Some of the more newer techniques for detection on chromatography systems, for instance, PDA or photodiode array UV or mass spec, can actually look outside of the focused area of the method and see if there are information about unknown peaks that could be showing up in analyses. And then, of course, we have this point about automating our calculations and removing data transfer. If things are automatically uploaded to a LIMS system for somebody else to review, then there isn't the option for people to hide or divert those results and replace them with better ones. And this, I just wanted to share a chromatogram. This is when a method that we took from HPLC, which runs in about 30 minutes, to a 7-minute UPLC method. But from my point of view, from a data integrity, the peaks are so much easier to integrate in the second example. Yes, you can get a 7-minute run instead of a 30-minute run, but what's most important is I can absolutely confidently integrate the areas of those peaks, separate them from the peaks nearby, and then I can have an automated method, which means as an analyst, I don't need to step in and make changes to the parameters in order to get accurate integration of those peaks. And that gives me a lot of confidence about right first-time peak integration. As I mentioned, for our impurities, we have to be sure that we're looking for all the peaks. The small ones, the insignificant ones, are we sure we can ignore them? Is there anything in there that we should really investigate further? Maybe look at a different detector method in case I can find something in using a different orthogonal detection. What are the reporting levels? Why has some of those peaks not been integrated? Or are they just not available on that particular wavelength that I'm monitoring, and they may be seen at a different wavelength? Do we have the right method for detection? It's a question we need to go back to our impurities tests and examine their scientific fit-for-use purpose. Now, this is an example here where using that MS detection can show me that where those two peaks that are really not very well resolved, I can look at the mass spectra of those peaks and say they are definitely two different compounds. They're not just a split peak. I shouldn't be integrating them together, and I'm probably going to very much overestimate impurity A because it's been contaminated in the chromatography with a lot of the API with a mass of 300. That extra information is enough to say, I put my hand up and I should say, there's something wrong with this analysis. I think I'm going to overestimate the impurity. I need to get a better method so I can separate these two peaks. And then looking at the calculation side of things, if we're doing things with calculators, it's far too easy to make a mistake. or to deliberately include some variation. If I'm using Excel, it's not protected. It has no Part 11. So any calculations done in Excel, if I could just change the number at the seventh decimal place, then everything would pass and I can go home, could be a cause of concern. So when we look at that, minimizing the human intervention, really automating as much as possible, having the software do as many of the calculations as possible is going to be advantageous. And that's only possible if the method that you're using is very robust. So these are very much tied together. Let's have a quick look at the third point in here, the courage to improve. So I talked about how methods can be improved, but quite often when we talk to people in the lab, they say, but this method is fixed. This method's been validated. This method's been registered. This is a monograph method. If I change it, if I improve it to make those peaks come apart so I can integrate it correctly, I'm going to have to do a whole bunch of validation. So methods do get very static. It's very important. At the moment, there are two initiatives which are really coming from the regulators saying, you know, if you can improve things in your laboratory, just as if you can improve things in manufacturing, you really should take the advantage to do that. And we shouldn't be putting burdens in the way to mean that you keep doing something that's inferior to rather than improving the processes. As I kind of hinted in that last session, the things that are labeled often data integrity concerns because of the analysts are actually much more about the method and whether the method is suitable for use. And there is a big push today about method modernization, but also life cycle management. I'm going to talk just briefly about those two things. I actually believe that we could solve a lot of the data integrity concerns by critically looking at the methods that we have and making sure that they're robust and fit for use. In fact, the warning letter that I pointed to earlier described in detail how, as part of the CAPA that happened to that company, they did consider changing the method to make it more robust, but they didn't implement that change. A rhyme in England called "There's a hole in my bucket," and this does kind of remind me of this story about I have this method, but I can't change it. You know, often labs say to us, oh, we have these concerns about peak integration, chromatographic peak integration, and our regulators or our external auditors are saying that we shouldn't be integrating peaks like this or reprocessing them or manually integrating the peaks. And they say, but with this method, I have to do that in order to get the right integration because the method isn't very good. The separation isn't good, it's noisy, it's got a rising baseline like I showed, or it varies a little bit every day with the temperature. And so when we say, well, why don't you develop the method, why don't you create a better method, the answer comes back, oh, it's registered with the regulators. We can't change that method because it's registered with the regulators. It's going to cost us money, we're going to have to get pre-approval to change a method. And then you know, the regulators are too strict, they won't allow us to change the method. So we go right round in a full circle. So we're trying to stop that circle and say, well, what point can we jump in and do something about this? And that's really where these couple of initiatives that are coming from the regulators are saying you should be allowed to improve methods if it's going to improve results. The courage to improve. Change can be seen as risky. People ask questions like, what about tests we already released? What about analytical methods that we use for stability? We've done the first part of the stability with one method. If I make a different method now, do I have to go back and reanalyze all the earlier samples? And one of the very constant things that people say is, well, what happens if this new method shows some new impurity that I didn't see using the old method? If I had a dollar for every time I'd heard that, I'd be a very rich lady. And this has been going on for a really long time. People are concerned that some new peak will show up and then there's going to be a whole bunch of paperwork and submissions required. What we learned from the nitrosamine crisis is that when a new peak does show up, somebody needs to be concerned. It is the time to raise your hand and do something about that. Registration of analytical methods is a problem because of the validation, qualification of a new instrument may be required. And the re-registration of those changes with regulatory authorities can be very expensive and very time-consuming. But then if we look on the other hand, what are the risks of not changing? we're probably going to be producing products with impurities which may need to be recalled from all over the world. The courage to improve is coming through an ICH process, which is looking at risk assessment and risk management, which is in ICH Q8. And we're awaiting the release of ICH Q12, which is a product life cycle approach to quality. This really is saying that when there is the appropriate time, we should be allowed to change parameters of critical manufacturing parameters within certain ranges if we have validated those as critical parameters and validated those ranges. And within that, they're also looking at what can we do about analytical methods. Can we qualify and register a space so I can make small adjustments without having to do a re-registration of the method? But typically as well, the method, before you even start using it, you need to stress test it to see, is it going to work throughout the life cycle of my product? Once that is out, there's a second group looking at a specific new ICH guidance on analytical method development to really encourage people to do more work in stage one, thinking about the method, thinking about its life cycle, where is it going to go, who's going to need to use it, and what the outcome of that should be. And once that has been released, ICH Q14, there's going to be a revisit of Q2, which is the analytical method validation, to try and apply those same risk and life cycle approaches. And if you want to know more, Waters have a campaign on methods.waters.com with lots of information about how we can really proactively improve analytical methods. As well as that, in looking at the software and the methods that people are using, the conservative nature of change, the FDA Medicinal Device Group are putting together a new guidance, Computer System Assurance, which we expect in 2020, which is really trying to say, if you can think of a way of improving things and computer system validation is your barrier, you should be looking at that with a risk-based approach as well. We're trying to get more innovation, trying to make sure that software can be updated, whether it's for security reasons or new features or better features, without being the stifled barrier of validation. So watch out for that guidance from FDA as well. So we really should have the courage to say, can I do these things better? Briefly talk about blind spots and what we learned from that from the nitrosamine process. As I said, a lot of the problems from this came just because there was a few places where the product was contaminated with the NDMA, but that contamination spread. And why did it do that? Well, we have a very complex supply chain at the moment, and product testing alone is probably insufficient. If you're testing your final product, if you think about that case, if somebody had tested the solvents before they were used, it would have been would not have needed a very sensitive method to do that, and that could have prevented the contamination. So incoming materials at every stage should need to be looking for potential, especially carcinogenic impurities, in-process checks to make sure they're not being formed. And if you're changing solvents or changing vessels or using equipment for different purposes, real good cleaning validation tests. And I've seen a big uptick in observations about people not doing very good or documented very well their validation, their cleaning and the cleaning validation to support that the cleaning is done properly. Almost every week I see an observation about somebody who was just relying on the certificate of analysis for their supplier, and that was just being sent to them and they believed it and didn't do any testing at all of it. Probably didn't even qualify that supplier very well. That quality oversight of your suppliers should be at the place where you would find out if there's an issue. And one of the other current trends I'm seeing at the moment, there's quite a lot of people who are using glycerin in pharmaceutical products, and glycerin is used in all sorts of food and over-the-counter dietary supplements. But the diethylene glycol contamination we know has been a real issue, and a lot of people are using glycerin, buying it in, and not having the assurance that there is actually no diethylene glycol in that product. When you are outsourcing, manufacturing, or insourcing product from other companies, you do need some oversight of the analytical work that is done at those sites, whether that's periodic audit or expert consultants that help you with that, or you have employees on that site, maybe in some cases. Are they sending all of their analytical data to you, or only the final selected results that give you the answers of the quality of those materials? do you have insight into all of the tests that they do? And if you're not sure at all from that point of view, then really you do need to do your own testing on incoming materials. I think it's just one of those blind spots that people have got used to trusting the manufacturers of the raw materials, and we really do need to be strong in looking for those, particularly impurities in those products. So go back to the reason why did this whole nitrosamine thing happen completely in the first place. It is really about critical thinking. That change in manufacturing process from whatever the solvent was before to DMMF, somebody should have realized that what could happen with that product is the creation of these nitrosamine impurities. So when there is a change in the manufacturing, we need to gather all of our chemical knowledge and our understanding, do a real big risk analysis for what could be here, because the analytical labs are only going to test what you tell them might be in something. So it's the production chemists that are going to say, there is a risk of this particular component being a byproduct of this reaction, and we should be monitoring for that. And during the process manufacturing change, we need to do hypothesis testing. Well, if we think it could happen, let's just see before we start manufacturing product if that happens. You may need to have there the very most sensitive analytical techniques because you're going to be looking to see whether there's even the faintest possibility of these impurities being created. Understanding the different ICH regulations about impurities, and I've included in here A, which is, I get this right, this is drug substance, B is drug product, and C, which is solvents, residual solvents that are left in materials, as well as M7, which is some very specific guidances about how to do the calculations of acceptable risks for DNA reactive impurities. What those guidances say, or what M7 specifically says, is prevention is much better than detection. Having a process that doesn't allow those things to create really is what we're aiming for. If there is the possibility, then we need to put the analytical techniques in place to detect those things. And so looking at impurities like nitrosamine, we have to look in detail at the process risks. What kind of things could be created using the conditions that we have in our manufacturing steps? Is there the possibility of carrying over from one step to another or one vessel to another? What are the event risks that could happen? What could be the incoming materials that might be causing a problem? What are the dangers of using recovered solvents versus fresh solvents? And what about the packaging? There's been some cases of contamination where material was found in the product that leaked from the inks on the packaging. That's the level of creative and critical thinking we need to think of when we're designing these things. Could this ink end up in the product somehow? And then in the area that obviously Waters we're thinking about is the analytical risks. Are people trained? Do you have an appropriate and qualified method? And do you have the courage as well there for people to actually say something's not quite right? So there were the 5 things that I think I learned from reading about the nitrosamine crisis and the remediation that we have to do. I think the biggest thing is that when people see something out of the ordinary in analytical results, they may be tempted just to hide it. Yeah, I'll just change it. It's not my place to say where this peak has come from. It's probably something I've done anyway. I don't really need to take any notice of it. I think it's important that we have to avoid that analysts have the motivation to polish results, whether it's because they don't have enough time and too many samples, they have old instruments or poor quality reagents. We're trying to save money, so we're not buying the top grade solvents maybe, or it's the methods which can't be changed. And I think there's a lot of that happens today. And management have to allow analysts to do their job, which is to notice and stop product being released from that particular process which doesn't meet specifications. There have been cases where management maybe defined what the design outcome is, maybe pass the batch as quickly as possible, and people will then be tempted to just do whatever management asks them to do. And also, we have this, I don't want to be blamed for doing the wrong thing. The whole concept of metrics, I've seen many labs where their metrics, their performance is based on how many batches you pass. So you're not going to put your hand up when something looks abnormal or unusual. it's a really tedious process if you do see something abnormal. And if it's not part of your metrics for your job, then it's very easy to kind of skip over that. And also, nobody will listen, maybe, or nobody will notice if I'm just cheating a little bit and everybody else is. Those are the kind of concepts that we need to avoid if we're going to be working in a quality culture. So I've talked about an awful lot there. I'm going to hand that back. I think it's a great time now to have questions.
Great, thank you very much, Heather, for a very interesting presentation there. It's now time for the question and answer session. So the first question we have here, what is the USP method for nitrosamine testing?
Yeah, most people would expect there to be a method for doing every kind of testing. And the USP really has methods for products or for excipients or for things that are meant to be in the product. So the nitrosamine testing is an interesting one, and we've been talking closely with the USP, and they are putting together a chapter specifically to call out what needs to be done for nitrosamines, how to approach that, because really we don't expect those nitrosamines to be in there. There have been some methods that have been put together by various vendors, experts, and some of the regulators as well. But as far as I know, the USP hasn't assigned what they think is the best method for this. So it's really up to people to look at the instruments they have, look at what levels they need to get to, and come up with an appropriate method for that. So I'm afraid to say there really isn't an USP method yet, but watch out, there will be something soon, I'm sure.
Great, thanks, Heather. The next question, what would be the right technology to quantify nitrosamine impurities? Do labs need high-resolution MS, LC, or GC?
Yeah, what's the right technology? Generally, whenever you want to do an analysis of a very targeted known compound, then you're going to use usually an LC or GC with a triple quad mass spectrometer. That's usually the technique of choice to go to for low-level quantitation of known compounds. It's what's used for any kind of screening test generally, or any quantitation of known pesticides will be done with that technique. And so that's going to give you the lowest levels of analysis that you can reach to. However, depending on where you are in the process, you may not need to get to that level. Obviously, if you're testing final drug product, you might need to be at the very most sensitive levels. If you're testing, the further you go towards the beginning of the manufacturing process, the higher the levels that you need to look at. So, for instance, testing solvents for NDMA would probably be done pretty simply with a GC or a UV kind of method. One other caveat in there is that the proposed source of the NDMA in the ranitidine product is actually a breakdown. It's not proven, but that's the proposed source of it. And when you put those kind of compounds, ranitidine, through a GC, it's quite likely you're going to be actually creating NDMA during the analysis. So GCs would not be the technique of choice for looking at ranitidine. So it really does depend what compound you're looking at, where in the process you are, and what levels of sensitivity you need to get to.
Great, thank you, Heather. The next question here, if the FDA or another regulatory body posts out a method, do I still need to validate it? Can I use my method if it's better?
I, again, would look at what I need to do, and if I have a method that is much better suited to the task at hand, or as the analytical QPD process says, the analytical target profile, I would be using the best method for the job. Just because even a USP method needs to be verified on your equipment in your lab. So if there was a USP method, you would still do some level of validation on that method. The methods that you can find by searching the different regulator websites, it's very useful that they've given you all the conditions they've used for testing those methods. But just because you see a method on there and just because that lab has validated, you probably do need to validate it in your lab on your equipment. Quite a lot of the methods we've seen on some of the regulated websites don't even use internal standards. And for an MS method, or particularly an MS method, an internal standard is really normally the most common thing that you would always do to ensure reproducibility. So I wouldn't take those methods and just run them without trying to improve them in my own lab and then validating them, because you really need to do the validation anyway.
Great, thank you. The next question, how could I know whether to trust a certificate of analysis from a supplier of my raw material?
Well, you could just see how lucky you feel. But yeah, if you know to trust it, really you need to have a very good confidence in the work that either that supplier is doing or whoever the lab or whoever that supplier has outsourced that testing to. They may be outsourcing that testing to a different lab in order to create the certificates of analysis. And there's just been so many horror stories around this recently. In one case, I seem to remember a case where they said that the salespeople were writing up the certificate of analysis in the car before they came in to sell you the product. We had a big case just in the US a few weeks ago where there was a lab that was manufacturing dietary supplements, and they'd outsourced the company, while the manufacturing, they'd outsourced the testing to a lab, and the lab said, we're not qualified to do GMP kind of testing. And yet those products were being put out and certificate analysis were being done from that lab, even though they weren't even had any awareness of regulatory requirements. So you really need to do proper supplier assessment. I talked about sending auditors, asking to that company to ask about their data integrity, about their methods and how they validate them, and about the qualification and the training of the people in the lab. Everything about how I can trust that data that's coming to me. And if that's the only supplier and you're really not happy with that quality and oversight, you want to get all the data they create, but you're probably going to end up doing your own testing as well.
Thanks, Heather. We've got time for one more question. And this question, how can I find out more about analytical method lifecycle approach?
Yeah, we're really hoping that these ICH guidances come out really soon. There are lots of people in the industry who are very involved. This is we're finally getting into the last mile of a very long race on this. It kind of started around 2004. So the process of those guidances is evolving, and what that actually is going to mean when it all gets released and then gets adopted by the regulations, we're waiting in anticipation. At Waters, as I mentioned, we have our website, waters.methods.com or methods.waters.com, I think. And if you go there, our pharmaceutical experts are keeping track of new and updated information, but we have a lot of interviews and podcasts as well by some of these experts who are talking about how we think this will change really the landscape of the laboratories and allow people to be able to move on and improve methods and not have to live with really bad methods where analysts are spending so much time trying to estimate the areas in chromatographic peaks. Whichever technique it is, they want to be able to improve methods so that we can get more accurate and trustworthy and robust results being associated with the quality of our product. That's a really good question. Thank you.
Great. Thank you, Heather. That's all we have time for today. So thank you very much, Heather, for today's informative discussion and presentation. And thank you to everyone joining us online and for your interesting questions. If you do have any other questions, please feel free to email me at editor@selectscience.net and I will follow up with your questions. So remember, you can also download a certificate of attendance in the related resources tab to the left of your screen. And if you'd like to listen to today's webinar again or to invite a friend to listen, it will be available to watch on demand in a few days' time. So goodbye and thank you to everyone joining us.